Hi everybody, this is Dr. John Pitts of the Centennial Schultz Clinic. We lead and invented much of the field of interventional orthopedics and regenerative medicine. Today I'd like to talk to patients who are interested in cultured stem cells, like what we do down in our Grand Cayman facility. So I'd like to try to explain in a little bit of medical detail, but still using layman's terms, something that's very complicated and complex in 5 minutes. Let's see how we can do it. So get a lot of questions on patients asking what's the number of cells they're going to have injected or grown out, etc. It sounds like a simple question, but actually is quite complex because you have to understand quite a bit of biology and how cells are grown, and then also understand that dosing for patients depending on their age, health, severity of problem, joint that's affected, what part of the joint we're injecting intra-articularly in the bone, etc., varies. And, there's not a lot of precise or great research that says, hey, this is the exact number of cells every person needs for this particular problem. So this is basically things we've learned over the years and will continue to change and adapt as we learn and research more and more. So first, I'd just like to start with the differences between having a same-day procedure and a culture cell procedure as far as just cell numbers. So, for example, if you do a same-day bone marrow procedure like we do in the U.S., you'll be given a total nucleated cell count, and that's the total number of cells and what's injected. That includes all kinds of different cell types, stem cells, hematopoietic stem cells. There are going to be some megakaryocytes and other types of different types of stem cells in that. And really the actual mesenchymal stem cell number in there is going to be in the tens of thousands. So 10,000 to 100,000, let's say, for example, to make numbers easy. If we culture cells out, we can get 100 to 1,000 times more of those actual stem cells, which we think are the most important cells. So we can get in the tens of millions or even in the hundreds of millions of total cells from one bone marrow drawn out. Okay, so even if we got a very poor yield of a stem cell, even if we only grew 1 million cells, which never really has happened, but that would be still 10 times more stem cells you get in the same day procedure. So more likely, realistically, the lowest you ever get is 100 times more stem cells with a culture procedure, and often it's mostly 100 to 1,000 times more stem cells. So that's one. And then two, is no direct way actually to measure stem cells in the same day procedure, whereas when you culture cells out, you can actually measure the cells more directly. Now, when you go through culturing, there's a several stage process. Okay, so when you obtain bone marrow, you have to plate that bone marrow and allow for the stem cells to stick to the surface that you place it on. We usually do this over a course of a 24-hour period, per se. Then you kind of get rid of the other cells, and then you can then transfer the stem cells that stick to the plate by releasing them and take them to other plates to grow out in much higher numbers. And every time they grow out, they run out of space and nutrients. You have to replenish that and grow them out again. So every time you do that, that's called a passage. So, typically it's not safe to grow cells beyond 4 or 5 passages. Okay, so typically when we do stem cells, we grow cells out to 3 to 5 passages. Now, we stop after 2 or 3 passages to freeze the cells, and then that way we can safety test the sample to make sure no bacterial or viral contaminants or any parasites or anything, just to make sure there's no infectious diseases within that sample to make sure everything is safe. And then when the patient comes back for their re-injection, we know when they're going to get their cells injected. So we pull those cells out of cryo slowly in a recovery culture over the course of 2 to 4 or 5 days, depending on what we need. And then we can grow those cells out another passage or so. And when the cells are delivered to the patient, they're coming fresh right off being grown in the lab. And at that point, we also do sterility testing, make sure it's, again, no contaminants. And then we also can do an exact cell count where we have the exact cell numbers. Now, sometimes we can estimate after growing them out for a passage or two how many we think we will have at the end stage, and that's how we kind of determine how many cells to grow and if we can store some, depending on the orders and the plan. But obviously that can vary quite a bit because cells don't grow at a uniform rate. It can vary quite a bit. And so typically you'll have your exact cell numbers right at the time of your procedure, because that's when we'll have a final count. We'll let you know if you have cells enough to store it over. And typically there's no issue with having enough cells, because there's a pretty broad range of desired target cell doses that you can use that are very effective to treat different body areas. And there's also, you don't want to just put as many cells as you can, because there's actually a limit to putting cells in where it's detrimental, not in the negative sense like you're going to cause harm, but where those too many stem cells, they don't fighting for nutrients, crowded out, not going to have a positive effect. So you only want to use that middle ground, the gray zone of having enough extra cells to get the job done, but not too many that you put in too many cells fighting for resources. So that's a brief 5-6 minute overview of how cells are culturing in numbers, and it's even more complicated than what I explained, but want to give you guys a taste just for your understanding. So when you're asking about cell counts and you're not getting exact numbers, this is why, because the whole process involved with that. You guys take care. Have a great day.